Welcome to Chapter 15 on our special senses with our flex point on hearing and balance. So first we're going to divide the ear into three different regions. We have our external ear, which will assist with hearing. It starts at the auricle, which is pictured over here, or also known as the pinna, where sound waves are going to funnel through into our external acoustic meatus and end at our eardrum, also known as the tympanic membrane. Next we have our middle ear, which is just this small section over here. This will also assist us with hearing and is an air-filled space where we're going to have our three auditory ossicles. And then lastly, we have beyond that point our inner ear, which is going to assist us with both hearing and balance. And it is composed of interconnecting fluid-filled tunnels and chambers within our temporal bone. You see major structures will be our cochlea, which is the snail shell over here, the vestibule, and our semicircular canals. You may also recognize our old friend cranial nerve number 8, which is our vestibulocochlear nerve. So we'll start off with the external ear. We mentioned the auricle or the pinna, which is composed of two different regions. We have the helix, which is the outer portion that is funneled or rim-shaped, and in the inferior region we have the lobule. Next we have the external acoustic meatus. This is just an auditory canal that will help those sound waves channel through all the way to our tympanic membrane, which we indicated in the previous slide. Within the external acoustic meatus, we are going to have it lined with skin, and we will have some hairs within here, some sebaceous glands, and a new structure, which are ceruminous glands. These are going to be modified apocrine sweat glands, and they produce a very special substance. They are going to produce cerumen, or what we know as earwax. So let's go to our tympanic membrane, which is that last portion of the external ear. This is our boundary and is made up of connective tissue that is going to assist in creating the vibrations from our sound waves and transferring the sound waves over to our three small ossicles that reside within the middle ear. So those sound waves, like we said, are being transferred into our middle ear. This area is air-filled. We should have no fluid in here unless there's some type of pathology taking place. But the walls in here are lined with mucosa, and below that mucosa is our temporal bone. We have our eardrum on the external portion, and then as we move more internal, we are going to see that there is a oval structure, which we are going to call the oval window. And so let's quickly talk about these bones and the way this sound is transferred. The tympanic membrane, just like a drum, is going to vibrate and cause the malleus, also known as the hammer, the incus, also known as the anvil, and the stapes, also known as the stirrup, to vibrate and pass that information along to this oval window, which will then displace fluid within our inner ear, which we'll get into soon. That fluid will continue moving all around, and eventually we'll see this region highlighted here by the orange, our round window. This little membranous area will displace a little bit as well in order to kind of dissipate those vibrations. Some other structures we have here is our epitympanic recess. This is just the superior portion of our middle ear. We also have our pharyngotympanic tube or auditory tube or eustachian tube. You should know all three terms for this. And this is a tube that we've discussed before in our digestive system. This is connecting our nasopharynx over to the middle ear. And the reason this exists is to help equalize pressure within our middle ear. So if you're going up in an airplane and your ears are popping, you might try swallowing something or yawning in order to get the pressure to equalize. This can also happen if you're going in an elevator into a high-rise building, basically whenever we get a change within altitudes. This is a review of our ear ossicles here. We said we have our malleus or the hammer, the incus or the anvil, and our stapes here. These are all suspended by ligaments and joined by synovial joints. We're also going to have two muscles to know in this region. Let me give you a view of these muscles. So once again, we can see our malleus, incus, and stapes. So this would be our anterior region, this would be the superior region, and this would be our lateral region. So think of your ear as being on this side of this magnified image. So first we have our tensor tympani muscle, which you see connecting over from our malleus. And then we also have our stapes muscle that connects from our stapes to our temporal bone. And these are going to work to contract reflexively in response to loud sounds to prevent any damage to our hearing receptors. Now let's discuss our inner ear. We are going to have three major structures that are encased in bone. So first we have the cochlea, that is the snail shell structure here, the vestibule in the center, which is in charge of balance, and also our semicircular canals in this region. And there are three different segments to this. We have an anterior portion, a posterior portion, and a lateral portion. And these are in charge of balance as well. And I forgot to mention that our cochlea is in charge of hearing on its own. Within these bony structures, we have membranous portions as well that are creating tunnels and chambers. And within these labyrinths, we are going to have certain fluids. The first type of fluid is endolymph, and this is found within the membranous labyrinth. So for example, here in the cochlea where you see the blue portion, this is known as the cochlear duct and contains endolymph. The other fluid that we are going to see is perilymph. And for some reason the word disappeared here, but this is going to be found in the space between the membranous labyrinth, which you see in blue here, and our periosteum of the temporal bone. So right in this region over here is where you would expect perilymph to be found. And so here's a better image of these different sections. So first I want to focus on this cochlea. When we look at the cochlea, you see that it looks like just one channel circling around. But if we were to take a cross section of this structure that looks like just one long tube wrapped up, it is actually three different regions. So the top region and the bottom region, these have specific names that we'll get into in a moment. But in blue, we can see is represented with perilymph, whereas the middle region that we discussed being the membranous labyrinth, known as the cochlear duct, is filled with endolymph. So endolymph is represented by the pink fluid here, perilymph is represented by the blue. Similarly, if we look at the semicircular canal, it just looks like one tunnel, but when we view it in cross section, we see that there is a membranous labyrinth that contains the endolymph, and there are several little pockets here within the bony labyrinth that are consisting of the perilymph in blue. So now let's take a closer look at those three regions within the cochlea and learn the names for those regions. So once again, I'm going to click quickly back to this view real quick just to describe what we're looking at. Here's our cochlea that we find within the inner ear, and that this cochlea in the next slide has actually been unwrapped. So I'm going to click back real quick. So this view down here, this is the cochlea untwisted so that you can see the communication within the different regions of the cochlea here. Let's look at the stapes first. We discussed that within the outer ear, we are taking those sound waves. Those sound waves are vibrating the tympanic membrane, which then sends those sound waves through the ossicles, the malleus incus, and the stapes. That stapes pushes on the oval window kind of like a pump and sends the vibrations, so I'm moving my arrow, creating vibrations here, and sends those sound waves or vibrations through this perilymph. That perilymph continues around, and as it's moving through, I want you to think of it as creating waves within this fluid. And when it does that, it causes this cochlear duct and endolymph within it to vibrate as well. So now let's take a look at this picture on top here. These are the three different regions in here. So here's the top, here's the middle, here's the bottom. So this is corresponding with the top, middle, and inferior region over here. Let's start off with our scala vestibuli. So the scala vestibuli is the one that was in contact with our oval window down here. Those vibrations were being sent through this region filled with perilymph. This then is going to extend all the way to the apex of the cochlea, also known as the helicotrema. And then we're going to send those waves down through that inferior region. This inferior region is known as your scala tympani. So there's our scala tympani. And as we're doing this, so we get sound vibrations that are moving up. If the sound waves are representing high frequency vibrations, those would be sensed down here in the lower region of the cochlea. Whereas if we have lower frequency vibrations, those would be experienced closer to the apex or the helicotrema. So then we continue on down, and eventually those vibrations are going to be displaced by the round window. So the middle region that is filled with endolymph is our scala media or cochlear duct. Now we said that as vibrations are being sent through here in the perilymph, the cochlear duct is being vibrated as well. And that happens because the tissues here are not rigid. If we look at the scala media here, you will see that it's bound by two membranes, one down here and one up here. The superior membrane is termed your vestibular membrane and is essentially the wall of our scala vestibuli, and that's where it got its name from, the vestibular membrane. Whereas the inferior membrane here is termed your basilar membrane and is essentially the wall of your scala tympani. Just as a reminder, your scala media or cochlear duct is filled with endolymph, which is in the pink. Now there's also one very special structure within your cochlear duct that assists us with hearing, and that is your spiral organ or organ of Corti. And this contains our special hair cells. So if you look at this picture below, it is a magnified image of your scala media. So just to orientate you real quick, all of this is our cochlear duct in here. Superiorly, we have your vestibular membrane. Inferiorly, we have your basilar membrane. And sitting on this membrane, we have your spiral organ or organ of Corti. Our organ of Corti contains hair cells that are going to essentially transmit sound. If we look at a close-up or a magnified view of one of these hair cells, you will see this hair cell in the light blue here, and at the very end, the apical region, we have microvilli that are called stereocilia. When we have sound waves move through and cause the endolymph within our cochlear duct to create waves, it pushes down on this structure known as your tectoral membrane. The pectoral membrane will then push upon the stereocilia on the hair cell, and that sound is transmitted through to our cochlear nerve that contains afferent fibers. So there are two different types of hair cells. We have our inner hair cells that are responsible for hearing, that will send fibers through our cochlear nerve. And we also have some outer hair cells represented over here. And these are connected to some supportive cells over here that lie upon the basilar membrane and are responsible for regulating the tension on our basilar membrane. As that sensory information is being transmitted to our cochlear nerve, they are going to have its cell bodies within this cochlear or spiral ganglion and then continue on its merry way to form the cochlear nerve. We're just going to touch upon some auditory function. We know that in order to hear, we have to create vibrations that produce sound waves. And we have different components that produce sound. We have the volume or loudness, which is a function of wave amplitude. So looking at this image over here, you can see that if we have lower volume, you will have these shorter sound waves, whereas if we have higher volume, then we have these longer sound waves. We can also change the pitch based on frequency. So if we look at this image at the letter C, we can see that if these waves are wider, or set up as lower frequency, we will get a lower pitch. Whereas if they are higher frequency or higher pitch, then we would see these waves are more narrow and closer together. Now let's put all of this together in order to understand where these sound waves are going. So here's our outer ear. This would be our external auditory canal where sound is entering. That causes a vibration to be created within our tympanic membrane. That's transmitted over to our malleus, our incus, and our stapes, which pushes against the oval window. From the oval window, we are going to cause vibrations within the perilymph of the scala vestibuli. This will continue through, causing vibration of the cochlear duct as well, which will vibrate the endolymph within the cochlear duct. This displaces the basilar membrane, which is seen over here. If we have short or high-pitch waves, we will have displacement of the basilar membrane near the oval window. If we have longer waves or a lower pitch, we are going to have displacement of the basilar membrane a bit further along from the oval window. So we can say maybe in this region over here. As the endolymph is moving, that basilar membrane is vibrating and the hair cells are being picked up by the spiral organ or organ of Corti, and those vibrations will also be transferred over to the perilymph in the scala tympani. Those vibrations will then be transferred to our round window here where they are going to dissipate. Now let's talk about neuronal pathways for hearing. We have our sensory information that we picked up via the spiral organ of Corti within the cochlea, and that was transferred through the cochlear ganglion and cochlear nerve over to our vestibulocochlear nerve. This is going to synapse at our cochlear nucleus within our brainstem. From there, we have an axon that'll extend over to the superior olivary nucleus within our brainstem as well. Or we can also synapse within our inferior colliculus. From there, we are going to travel to a special nucleus within the thalamus. This is termed the medial geniculate nucleus. And from there, an axon will carry that sensory information over to our auditory cortex within our temporal lobe. Now that information that was sent over through the vestibulocochlear nerve over to our superior olivary nucleus can also synapse with some efferent fibers seen in red here that will head over to our inner ear or to our motor nuclei within the brainstem, to our middle ear muscles that are going to be responsible for contracting reflexively if we hear loud sounds to prevent any damage to our hearing receptors. Now let's talk about balance. Within our vestibule, we have two important structures. We have our saccule in our vestibule and also our utricle within the vestibule. And this helps to evaluate the position of our head relative to gravity. It's going to help us to detect linear acceleration and deceleration, as if we were in a car or if we are in an elevator moving up and down, or if you're in an airport and stepping on one of those conveyor belts to kind of accelerate you forward as you're walking. So we have a kinetic labyrinth as well. So this utricle and saccule is responsible for static equilibrium. So we call this our static labyrinth. Whereas our semicircular canals that we mentioned at the beginning of this lecture, they're responsible for kinetic or dynamic equilibrium. So we are going to call this the kinetic labyrinth. And this helps to evaluate our movement of our head in more of a three-dimensional space. So if you are on a roller coaster spinning in circles, things like that. So to go into detail about static equilibrium, first we'll discuss the utricle. Within the utricle, we have macula that are going to be parallel to the base of the skull, and the saccule is going to be perpendicular to the base of our skull. And by having it arranged this way, we can differentiate between the static movement. What is a macula? The macula is going to be specialized epithelium that has these columnar cells seen in purple over here that are going to contain hair cells with lots of stereocilia and one cilium or kinocilium that is embedded within a gelatinous mass weighed by otoliths. Otoliths, think of it as like a stone. So the stone that you see pictured here is going to move or weigh more on one side than another, and that will help determine where we are in space. Here's a nice image that depicts that. So here is this man just standing head upright, and you could see all of the kinocilium and our stereocilia are all lined vertically. And so as the man bends over to pick up this box, those otoliths on top are going to weigh the gelatinous membrane in one direction, which bends these kinocilium and stereocilia so that we can see that there's a displacement within this macula, and we know that our head has changed its position in relation to what it was before. So now let's talk about our kinetic labyrinth. We have our three semicircular canals that were filled with endolymph. We have our anterior, posterior, and lateral portions of our semicircular canals, and they are placed within our transverse plane, the coronal plane, and sagittal plane. At the base of each of these are going to be expanded into ampulla, and this is going to have sensory epithelium, and you could see at the bottom portion of this image we have this sensory epithelium, and we term this crista ampullaris. And on top of this crista ampullaris, we have the cupula. The cupula is suspended over the crista hair cells. And as we see displacements of these hair cells, we know that our position has changed. So here's a nice image that depicts that. Here's our cupula, here's the hair cells, and this would be representing our crista ampullaris. And so if you are a figure skater and you start spinning in circles, there will be a displacement of this cupula, which will also displace the hair cells. And now the last thing we are going to discuss is the neuronal pathway for balance. We picked up that sensory information for balance from our semicircular canals as well as the utricle and saccule within our vestibule, and we are passing it through our vestibular nerve. We also, just so you know, have a vestibular ganglion where the cell bodies are going to reside that belong to the axons, carrying the sensory information through to our vestibulocochlear nerve. We are then going to synapse within a couple different places because we are going to integrate a lot of balance information and send it to various locations as well. We are not only receiving sensory information of balance within the inner ear, we are also receiving balance information from our proprioceptors within our muscles. So areas from our legs are also coming over to this region over here known as our vestibular nucleus. We are then going to send information to our cerebellum, which will influence our postural muscles. We've discussed in the past in our brain lecture that the cerebellum helps a lot with posture and balance. We are also going to send some neurons over to our motor nuclei that will influence our oculomotor nerve, our trochlear nerve, and our abducens nerve to control our extrinsic eye muscles, and also send axons over to our thalamus in a new nucleus. This one is termed the posterior ventral nucleus of the thalamus and travels over to our vestibular area of the cortex.